Ever wondered how your cells copy billions of DNA letters so precisely? Stick around, and we'll break it down step by step. DNA replication is the process by which a cell makes an exact copy of its DNA before it divides. Every cell in your body has the same DNA, and this replication process ensures that new cells get an identical copy of the genetic material. Replication is crucial because, without it, cells wouldn't be able to divide and create new ones. Whether it's for growth, repair, or reproduction, DNA replication is happening constantly in your body. DNA structure. DNA is made up of two strands that form a twisted ladder-like shape called a double helix. The sides of the ladder are made up of sugar and phosphate molecules, while the rungs are made of nitrogenous base pairs, adenine, A, pairs with thymine, T, and cytosine, C, pairs with guanine, G. These base pairs are what make up your genetic code, and during replication, each strand serves as a template to create a new strand. The steps of DNA replication. There are three major steps: initiation, elongation, and termination. Step 1. Initiation. The first step is initiation. Replication starts at specific points on the DNA, called origins of replication. An enzyme called helicase unwinds the double helix, breaking the hydrogen bonds between the base pairs and creating two separate strands, forming what's known as a replication fork. These separated strands now serve as templates for the new DNA strands. At the replication fork, proteins and enzymes gather to kick off the replication process. Step 2. Elongation. An enzyme called DNA polymerase is the star of this stage. It attaches to the DNA template strand and adds complementary nucleotides to form a new strand. If the template strand has an adenine, A, DNA polymerase adds a thymine, T, to the new strand. If it has a cytosine, C, it adds a guanine, G, and so on. This process happens in two directions. The leading strand is synthesized continuously in the same direction as the replication fork. The lagging strand is synthesized in short, separate fragments called Okazaki fragments, since it runs in the opposite direction. These fragments are later joined by another enzyme called DNA ligase. Step 3. Termination. Once the entire DNA molecule has been copied, the newly synthesized strands are proofread by DNA polymerase to correct any errors. Another enzyme, ligase, then seals up any gaps between the Okazaki fragments on the lagging strand, making it a continuous strand. At the end of the process, we have two identical DNA molecules, each with one old strand and one new strand. This is called semi-conservative replication because each new DNA molecule conserves one of the original strands.